registered in 2018. We have R&D offices in Bangalore, Raipur, and Bremen, Germany. Backed by Tata Social Alpha, incubated at 𝗃𝗃 INC and IIIT Hyderabad. We have developed 𝗪𝗏𝗡, which is a patented automatic sperm washing centrifuge. And the main goal of 𝗂𝗏 is to help the gynecologist set up IUI services. So our special focus and attention is in setting up IUI labs. 𝗂𝗏 also featured in Shark Tank India Season 2 in March 2023, where we got a deal from Namita Thapar. And this is the leadership team headed by Vikram Rajput, who lives in Germany. Then me, I take care of operations. Then Chandan, who is taking care of marketing. And we have Dr. 𝗬𝗏 who is our medical advisor. Okay, so coming straight to the topic, what is sperm washing? So this is the definition given by WHO. So the separation of human spermatozoa from seminal plasma to yield a final preparation containing a high percentage of morphologically normal and motile cells, free from debris, non-germ cells, and dead spermatozoa is called sperm washing. So basically, it's the separation of sperm from the rest of the semen, if we see in the normal terms. Okay, so what exactly happens when we do sperm washing? So the target is to separate the sperm, basically healthy sperms, but in the process, there are harmful elements present in the seminal plasma, which we need to remove. So one is, and most important is removal of prostaglandin. So if this goes directly into the uterus, this will cause severe uterine cramping. So this has to be removed. So we cannot do IUI without washing the sperm. So that's the very basic requirement. Then the next is removal of dead and abnormal sperms. If we don't remove the dead sperms, then the dead sperms release reactive oxygen species, ROS, which actually damage the DNA of the live sperms. Then next is the removal of leukocytes or white blood cells. So again, here this element, when this is present, this actually generates the reactive oxygen species, and in turn it damages the sperms, the live sperms. And then the next is removal of bacteria and viruses. So if that goes directly into the uterus, it will cause infection, and we have to be especially careful about the HIV virus. So by doing sperm washing, we are also removing the virus like the HIV virus. And then another important factor is the decapacitation factor. So basically, these are the proteins and enzymes which prevent this sperm from prematurely getting activated. Before the sperm can fertilize the egg, it needs to go through the capacitation process. So basically, the removal of these proteins and enzymes actually ensures or enables the sperm to get capacitated. So that's, and then last, the very, very important factor is especially in case of IUI. Typically, we say that semen volume is normal when it is 1.5 ml or more, but we cannot inseminate the entire ejaculate volume into the uterus. So, and so the typical normal volume is, let's say 0.3, 0.4 ml in the normal condition. So we can inseminate only up to that volume. So from the total ejaculate, if we just take a portion of it, so ultimately only the portion of the total count will go into the uterus. Accordingly, the chances of pregnancy will be reduced if we are not able to inseminate all the sperm into the uterus. So basically, here by separating the sperm from the rest, we are concentrating the volume. So what are different methods of sperm washing? So the very basic method is known as the swim-up method, where we take the liquefied semen. So the first step for any sperm washing is collection of semen. As you might all know, in a collection jar like this, sterile jar. So the donor, basically the husband, collects the semen into this. We put the cap, and then we keep this in either room temperature if it is close to 37 degree, or we put this into either warmer, which is set at 37 degree, or the dry incubator again at 37 degree. And depending upon the viscosity of the sample, this may take up to 30 minutes or even more than that. But the recommended time for liquefaction is roughly 25 to 30 minutes. That will happen when we are doing the normalization in at 37 degree. So semen collection, liquefaction, and then there is a sperm washing. So in this swim up method, which does not require centrifugation, we take the liquefied semen. So I'll go into the details of the process, how various steps are done in the subsequent slides. Here I'm just giving overview. So the liquefied semen we take into the tube, a normal centrifuge tube, and then we layer culture medium over it, and then we keep it at 37 degree, 45 degree in 37 degree temperature. And typically it may take up to 1 hour to get these sperms migrate into the culture medium. And post that, we separate this layer into pipette, transfer into the tube, and then we take it for the further process. And then on the other side is the centrifugation method, where there is a simple wash and then there is a density gradient method. In simple wash, what we do after liquefaction, so any process of sperm washing starts after liquefaction. So after liquefaction, we mix culture medium and then we put that into centrifuge, and then we centrifuge it. In the process, the live motile sperm get deposited or get pelleted at the bottom of the tube, and then we discard the supernatant. And then typically any centrifugation is a two-step process. We first centrifuge it, discard the supernatant, then mix the culture medium again, put it back, centrifuge it, and then again discard the supernatant. Then the final pellet gets ready, in which we again mix the culture medium or buffer so that we prepare the inseminate volume. So the simple wash is we are mixing culture medium, centrifuging, and then the pellet is getting ready. In the density gradient method, we have, so let me just show you the media. So in simple wash, we have this media, which is culture medium. Only this is used. In the density gradient method, we have this set of media. So basically low density, high density, and culture medium. So we make a layer of high density, as you see here, high density, low density, and over that there will be semen, liquefied semen. For making layer, there are different methods. And so this androwash system specialized in making the layer very easy, quick, and accurate. So I'll be talking about this in subsequent slides. High density, low density, and liquefied semen. And then we put that into centrifuge and then centrifuge it. Again, there will be pellet formation like this here. And then, so how different or how better is this process as compared to the simple wash is, as you can see, the density gradient. Basically, what we are doing is we are creating these interfaces. Here, everything is mixed. So there's no distinct layer or interface where we can separate different elements of elements present in the semen. So in the density gradient, what happens is here the seminal plasma gets filtered out. It cannot travel below this. And then the dead sperms get separated here. And only the live sperms come here. So this way, this is considered the best method of sperm washing. Here, the swim up method is basically based on the assumption that the sample has a good amount of motile sperms. Otherwise, if there is no motility, then we'll not get the sperms into this layer. And this method is actually not able to remove the leukocytes. And even the dead sperms are present. It doesn't fully remove the dead sperms also. That's why this method is typically not used in case of IUI or even IVF. This may be used, or usually the embryologist prefers simple wash for ICSI. But for IUI and IVF, if you ask me what is the best or default method, then the density gradient is the one. Because this is the method which can give you the best, like the contamination free, most contamination free, and the best possible recovery. So again, it's a two-step method. Pellet formation happens after the step one. Remove the supernatant, again mix the culture medium, put it back into the centrifuge it, and then again discard the supernatant. Then to make the final inseminate, we mix the culture medium properly, and then we load that into cannula. So going into the details of the process. So typically when you hear from someone about how to do sperm washing, so people just say that you centrifuge it at someone will say that you do it at 1000 RPM, or some other will say 1200 RPM, 1500 RPM. So it's actually not the RPM which is important. As you can see, it's the G value. So going step by step. So first as we see, this is the first step, liquefaction, and then we mix the sample first by pipetting, and then we dilute uh, the sample 1:1, and then based on the total amount, if it is more than 3 ml, then we split that into number of tubes. Because if we put everything in a single tube, so the separation will not happen properly. And how we should do is, for how much time, what RPM is this? This is the guideline. So this is the WHO guideline. So it says that 300 to 500 G. And what exactly is this 300 to 500 G? So basically it's a RCF, relative centrifugal force, for which there is a formula. RCF is equal to R omega square into 1.118 into 10 raised to power minus 5. So we need to know the radius of the centrifuge and the RPM at which we are operating. Then only we can calculate the G value. And we should calculate the G value, and then only we fix the protocol. So it says 300 to 500 G for 5 to 10 minutes. And then after that, so there is a pellet, then there is a supernatant. So we discard the supernatant. Basically, it's talking about that carefully aspirate and discard the supernatant. And then after that, resuspend the combined sperm pellet in 1 ml of. So maybe we have split that into multiple tubes. So after step one, we combine everything and then resuspend it. And then again we put it back into the machine, the centrifuge, process it. So here also the G value is 300 to 500 G only. Time is different. In step one, it's 5 to 10 minutes. So some people will centrifuge at 15 minutes. So that's usually it's not recommended. So we have to follow the WHO guideline. So 5 to 10 minutes for step one and 3 to 5 minutes for step two. And again, after the centrifugation gets completed, we need to aspirate and discard the supernatant. And then the final pellet, resuspend this pellet by pipetting. So this we are preparing for the insemination. So particularly we are talking about IUI here. So in IUI, we inseminate maximum up to 0.4, or in rare cases, we go up to 0.5, but not more than that. If so, the pellet volume is typically 0.2, 0.25. So the difference to basically make it 0.4, we need to mix the culture medium or buffer. And so basically it's explain that only resuspend the pellet by gentle pipetting. So once we get the pellet, we take the culture media with the help of pipette and do the pipetting so that it gets the sperms get dispersed properly in the culture medium or the buffer. Swim up method. So again, we mix the sample, then we take the semen in a conical centrifuge tube, and then over that we layer culture medium 1.2 ml. So this is a WHO guideline. So but individual lab may have their own protocol, but try to be close to the process defined by the WHO. So we, as I explained here, this is the semen, liquefied semen, and then we are layering the culture medium. And there is a swim down method also, in which case it is reverse. And after layering, we incline the tube at 45-degree, and then so basically the purpose of keeping it inclined is to increase the interface, and then we incubate it, or basically we keep it at 37-degree for 1 hour, and then gently return the tube to the upright position and remove the uppermost. So 1 ml, the top 1 ml layer will be removed, and then dilute this with 1.5 to 2 ml of supplemented again culture medium. So this is not a pure swim up method. Basically, it's swim up followed by centrifugation. So the basically the in pellet or whatever we say, which we have collected. So we need to centrifuge that for 5 minutes, and then we get the pellet ready. Then comes the density gradient method. So in density gradient method, so as I said, low density, high density. So typically we see 40-80 or 45-90 combination. Both are okay. So here also after liquefaction, we are mixing the sample, and then we make the layer of the gradients, and then over that we layer the liquefied semen. So here again the guideline is this way. This is a typical guideline recommended in the volume 5. Now it actually says that the manufacturer can define a protocol, the scientific manufacturer or whoever gives the sperm washing solution, or the lab can set up their own protocol. But this is a typical recommendation. So 300 to 500 G, again, we have to keep in mind that 300 to 500 G. So there is a recommended G value. Again, here is not talking about the RPM. So two-step method: make layer, put it into the centrifuge it, then the pellet gets formed, discard the supernatant, again mix the culture medium, put it back into the machine, then again there will be a supernatant pellet, so discarding the supernatant, and then we get the final pellet. So if we see what is positive and what is negative in different methods. So simple wash, what is the positive side? So it's very simple and fast. The consumable cost is also very minimal, and this is useful when the sample quality is normal. Or basically, it technically we call normozoospermic samples with high sperm counts. And what's the downside of it? It does not effectively separate the motile sperms from the debris or immotile sperm. The final the pellet may include dead sperms and contaminants, and the lower sperm quality, it's not usually recommended uh, to process in the simple wash method. In swim up, what is the positive side? It gives you the highly motile sperm. The method is very, if it is a pure swim up, then even you don't need a centrifuge. But pure swim up has a very limited application. It's very simple, less time consuming, no specialized equipment like centrifuge you don't need in case of pure swim up. And this is the method which gives you the least possible DNA damage because we are not centrifuging it. So uh, in cases where we get multiple failures, miscarriages, post DNA analysis, if we find that the DNA fragmentation is high, so typically we don't go for the centrifugation method. There we have to use the swim up method. And what's the downside? The recovery rate is very less because this method actually does not increase the motile percentage, unlike the centrifugation method. May miss some motile sperms trapped in the seminal plasma. So as, for example, simple wash, we are mixing, so the trapping gets released. So that's why we get a higher recovery. Less effective for samples with low sperm motility. So as I said, the assumption here is the sample has a good amount of motile sperms, then only we go with swim up method. Then coming to the density gradient method, higher recovery of motile, viable and morphologically normal sample. So this is the method which gives you the highest recovery. Then effective for low sperm count or poor motility samples as well. Reduces sperm deformities and debris. And what's the downside? It's more time consuming as we have to make layer, and the consumable cost is also high and may recover immotile sperms, potentially lower DNA. So this method also causes DNA damage. So like any centrifugation method will cause DNA damage. So what are the key factors which impact the result or which are important while doing sperm washing or while selecting the sperm washing? The very basic is the quality of media. So we should use the trusted brand, the validated ones, because uh, we there are many make available in the market. All of every media may not be of good quality. So before we use that, we should test that pre-wash, post-wash, and then only we should uh, finalize any media. So the selection is very, very important. And then ensure cold chain transport of media from the manufacturer to the clinic. So many suppliers, they don't ensure the cold chain transport. So that's why we need to be very sure that from whom we are buying and whether the manufacturer understands the importance of that. Many times we hear that, particularly in case of IUI, the manufacturers, they don't pay attention to the transporting, the cold chain transporting. So sperm washing is sperm washing, whether it's IUI or IVF. We cannot compromise on the quality aspects of it. The storage temperature, once it reaches to your clinic, you have to store that in 2 to 8 degree refrigerator. Basically, it's uh, usual household refrigerator is also set at 2 to 8 degree. But in hospitals, you have the vaccine storage refrigerator. So we don't have to keep it in the freezer. It's the refrigerator compartment, 2 to 8 degree, where we have to keep it. And then this is very critical point, bringing media to the room temperature, that is 37 degree before using this sperm, using for sperm washing. So this can be either done by keeping it in the normal ambient temperature, or putting this into the dry incubator or the warmer. So this is very, very important. I would like to develop on this a bit more. Uh, so how to ensure that we bring the media to the desired temperature in right time? So some manufacturers recommend that you take out the media from the refrigerator 2 hours in advance before you use that in the sperm washing process. But uh, the sample gets liquefied typically in half an hour. So let's say if you have taken out the media from the refrigerator, you have planned, but let's say the patient doesn't come, then what will happen? Your media will be wasted, because once it comes to the normal temperature, so either you have to use it or discard it. And even if you do or take out the media from the refrigerator after the patient comes, but there are chances that the patient is not able to give you sample, the semen. So then also the same thing will happen. So we have to synchronize the process. So how we synchronize it? When we have either dry incubator or the warmer. So once the sample is collected, we take out the as soon as the sample is collected, we take out the media from the refrigerator and we put media and sample into the warmer or the dry incubator. And so simultaneously, media as well as the semen sample come to the normal temperature. So this synchronization is very, very important. Many times we see that some clinics, they don't allow enough time, so that actually can cause thermal shock for the sperms. So that's absolutely not recommended. Sample collection, semen sample collection, we have to give clear instructions to the uh, basically the donors, the husbands. They have to maintain the abstinence period 2 to 7 days. This is typically a WHO guideline again, uh, but there has to be minimum 1 or 2 days abstinence period. The collection jar has to be sterile. While doing semen analysis, some labs use non-sterile jar. That for analysis that may be okay, but for sperm washing, for IUI or any ART procedure, we have to use sterile jar. Then collection of complete ejaculate. We have to make sure, or we have to give a clear instruction to the husband that collection has to be complete. Like they should not miss the initial ejaculate, which is actually very rich in sperm. And next is liquefaction. So as I talked in the previous section also, liquefaction is very, very important. So again, either so If the temperature, ambient temperature is close to 37 degree, then maybe you don't need warmer or incubator. But preferably to standardize the process, to synchronize the process, we recommend using warmer or the dry incubator. And another thing which is not mentioned here is in some cases, so typically the collection is done by masturbation, but sometimes if that is not possible, then we have to suggest using non-spermicidal condom if that is the method for sample collection. Then next is layering. Layering is very, very important, especially in case of the density gradient method, layering of high density, low density, and the semen sample. So how good we make the layer, that ensures the recovery as well as uh, the level of contamination present in the pellet. So if we do good layering, better layering, we'll get better results. Then centrifugation. In centrifugation, there are two parameters which are important, as I talked earlier. G value, which is RCF, relative centrifugal force. So we need to know at what RPM, what is the G value coming. And the G value has to be within the recommended range of 300 to 500 G. If so, neither less G value nor more G value than the recommended is suggested, because if we go with lower G value, so either recovery will not happen or it will take longer time, and taking longer time is not recommended, because as we delay the process, the ROS generation actually increases, the reactive oxygen species generation increases, which in turn damage the DNA of the sperm. So we have to do the processing, basically the washing, as well as the insemination as soon as possible. And then discarding is also very, very important. The discarding has to be precise. Otherwise, if discarding is not proper, then the final pellet will have the contaminants, the dead sperms. So discarding also has to be very precise. So how should be the ideal sperm washing technique? So it should be quick, easy, and cost effective. It should isolate as much motile sperms as possible. It should not cause sperm damage or non-physiological alterations of the separated sperm cells. It should eliminate dead sperms and other cells, including leukocytes and bacteria. Should eliminate toxic or bioactive substances like decapacitation factors or reactive oxygen species. Allow processing of larger volumes of ejaculate, and it should be repeatable and consistent. So this I have already talked in previous slides. Abstinence period, especially I was talking about this when the collection is through intercourse. Non-spermicidal condom. We can get this from the pharmacy, but we have to specially ask for this, and then we get it. Labeling is also very, very important. Otherwise, there are chances of mix-up if you are processing multiple samples at a time. So that's very, very important. And then the first portion of ejaculate should not be lost. And this is the typical threshold values for different parameters. So for the semen volume, it's 1.5 ml. So when it is 1.5 ml or more, we say it's normal. And then the total sperm number, that's the total count, should be 39 million or more. And here is a concentration. So many technicians or many labs get confused between these two. So concentration is million per ml and it is million. So concentration multiplied by volume, we get the total sperm count. So concentration, what's the acceptance criteria? 15 million per ml or more. Then we say it's normal. So these parameters are very, very important. So unless we measure it properly, unless we report it properly, we'll not do the right treatment. Then the next is motility. So it's progressive and non-progressive. Total should be more than 40%. And if we talk individually about the progressive motility, so it should be 32% or more. And then there are other parameters also. And then morphology. So I have seen many reports in which they mention 90% morphology, 80% morphology. So whenever you see reports like that, you have to doubt whether this the interpretation is correct or not, because when WHO says that 4%, close to 4% you get in case of normal samples, so there are highly less chances that we get morphology, 90% morphology. So what's the acceptance criteria? 4% or more is the normal morphology. And then pH level. So these are the basic parameters which we should definitely check and report properly. And based on what kind of abnormality we see in the sample, we we nominate it in different ways. So I will not go line by line with this. If anyone is interested, so reporting or using the right nomenclature is very, very important to interpret the result and accordingly decide the treatment protocol. So these parameters are especially important in case of semen analysis. Why I am stressing upon this is when we are doing sperm washing, we do pre-wash and post-wash checks, and accordingly we decide. So basically, all these exercise should be done before we decide whether we can go for IUI or we should go for IVF or ICSI. So this is actually a part of your diagnosis process. So here I stop. Now we can take the question answer. I'll be happy to answer your questions. So you, Chandan, you can allow the participants, so the participants can raise hand and ask questions. So
Hello,
either
everyone.
put your questions in chat box or you can unmute yourself
Yeah, so I have
and
given
ask.
you to unmute yourself. So if you have any questions, so you can unmute and ask the question. And if you don't want to speak, you can raise your hand and you can uh, share your question in chat box also. Thank you. By the way, Sohan, we have two question. No, one question. Can we use bicarbopher instead of culture media in case of IUI? From Isha Rajput. You can see the chat box.
Yeah, let me go through it. Okay, let's go through the question one by one. Second method is density gradient. In case of IUI, yes, that is acceptable.
Okay.
So either we use buffer or culture media while preparing the final inseminate.
Okay. So do you have any another question from anyone? please share your question in the chat box, or if you have a question you want to speak, I'm allowing you to speak here.
Hi, sir.
Hi,
This is
Viresh.
Viresh.
Yeah.
Sir, actually, now we are following 6th edition or 5th edition WH.
It's 6th edition.
But what you shown that was 5th edition now.
Right, right. So, as I said, for example, the threshold value, now the 6th edition does not give you a threshold value, but still that is used as a reference to decide whether this is normal or not. And the reason why that threshold values are removed from the manual is because that is actually not a representative values for because different ethnicities have different values, different observations. So that's why they removed it. But still, that is good to use as a reference.
And one more, sir. We can segregate the from DG for DNA damaged cells. We can segregate.
So can we segregate? That's your question. So so typically, like when we are doing a normal semen analysis, routine semen analysis, we typically don't see, or we don't go for DNA fragmentation test, right? DFI test. So by default, we are going with density gradient method. But let's say by any chance, or maybe let's say there is a multiple pregnancy failure. In that case, we are doing the DFI, and then we get to know that there is a high DFI. So in that case, it's not recommended actually, because any centrifugation method will only increase the DNA fragmentation. So if possible, if we get a good, let's say by swim-up method, we have to go with swim-up method.
Yeah, sir, that was okay. And
Yeah.
if you select the swim-up method now, in that we can get DNA damage from cells, so we can get from the supernatant, right?
Correct. So then if you are very particular about not getting DNA damage sample, then we have to go with advanced method. So basic methods will not be able to actually give you DNA damage free samples. So that requires an advanced method. Basic method will not give you the desired result.
Okay, sir. Thank you.
Yeah.
Thank you, Viresh. So any other participant who have any question regarding a sperm washing, so they can ask. If they want to speak or they want to share in chat box, they can share that.
Hi, sir. This is
Hi.
Arpita. Once sample is ready, the minimum how much time should we wait, or what is the good time to inseminate?
So there's no any such guideline, but we can wait for 5-10 minutes, not more than that. And in fact, it is recommended that we inseminate as soon as possible, because as we delay, so actually the sperms start dying. So when you process the sperm, you actually reduce the possibility of ROS generation. But as the time passes, more sperms will die, and in turn that will create ROS. So we shouldn't delay it. In case of IVF, we preserve it, we incubate it. So in that case, it's okay. But in case of IUI, we actually don't incubate, right? So we have to inseminate as soon as possible.
Okay. Like within half an hour or within 15 minutes, not like that, no, sir? Like.
So if you ask me, the answer is as soon as possible. Don't delay it unnecessarily. So we have seen some labs doing insemination after 2 hours. How will you get success? And then they complain that we are not getting success. So we have to just keep in mind as soon as possible.
Oh, okay, sir.
Okay.
Thank you, Arpita.
Okay. Then, sir, that sperm wash media, that culture media we are using, no? What is the stability for that, sir? Even that media that we are using for a DG.
Right. So, so there is a shelf life defined for this. Okay. I think I have not mentioned that in my presentation.
Yes,
so
sir.
in the pack, you see the expiry date of this. Okay. And then there is a color change also. But again, you have to be very careful, or you have to be like very vigile to see the color change. Otherwise, simply what anyone and everyone can follow is the label. It talks about the shelf life, the expiry date. So simply first check that and then use it.
Okay, sir.
Okay.
Thank you, sir.
And so for validation, you can check the pH value.
Okay, sir. Okay, thank you.
Okay, so another question from Rishabh Verma. Preservative using in during collection and storage temperature.
Used during collection. Maybe if you can elaborate the question, so I'll be able to answer that. So typically, if I have understood your question, you are talking about preservative. So for collection as such, there is no preservative used, and storage temperature for if you're asking for media, it's 2 to 8 degree, and semen sample for liquefaction, it's 37 degree. So it's actually not a storage temperature, it's a liquefaction temperature. If you can unmute yourself and elaborate the question, maybe we can have a better discussion on this.
He said, okay, okay. So again, from the question, Rishabh Rajput, in some cases,
while doing, yeah, let me go through this. Chandan.
Okay, yeah.
So in some cases, while doing DGC, after centrifugation, sometimes the pellet gets collected on the lower wall of the tube instead of collecting at the bottom. What to do in that case? Okay, so sometimes this kind of problem comes. So maybe we have to check the parameters and recheck the centrifugation parameters. Ideally, when we are using the recommended G value, this problem should not come. And just in case, if you get this, then discarding is very important. So you have to be careful about the discarding. Otherwise, if you are getting a pellet formation, so okay, but then be careful about discarding it. Otherwise, the first checkpoint is whether we are doing or we are following the right centrifugation parameters or not.
Okay, other question from Prajwal. After DG, we go for swim up is good or not?
Yeah, so that is pellet swim up. That is absolutely okay. You can go ahead with that. Some cases samples have more liquefaction time. In that case, what will we do? So like let's try to follow the standard method, like putting it into warmer or dry incubator. And even if that is taking time, so we can mix culture medium so that may expedite the liquefaction.
Hi, sir. Alina here.
Hi,
So,
Elena.
what is the need for 2-time centrifugation in simple wash? Because sometimes in simple wash, also 1-time wash is followed, right?
Right. So why two-step? Basically, so in step 2, we are actually, let's say, diluting the contaminant because pellet formation has already happened. So be it simple wash or the DG method, pellet formation is happening after step 1 only. But then there are some contaminants left over. So either, so as a result of improper supernatant discarding, or even if you do discarding properly, there may be some leftover because this is not a very precise method, right? So there is always a possibility of contaminants left over. So we try to dilute that. We try to minimize the presence of contaminant. So that is the reason why we go for second step.
So one more thing, sir. So would there be any difference in any parameters we are checking in the semen if we are deciding to do DG versus 2-time simple wash?
Can you repeat the question?
should there be any specific criteria that we are looking in the semen parameters before we are deciding if we should process it in DG or simple wash using 2 washes?
Yeah, so if we are talking about IUI, so as we saw, simple wash is not effective in removing the contaminants. So better method is DG, and we may go for the simple wash when the parameters are normal. But typically, when we plan or when we talk about IUI, we assume that the sperm parameters are not normal. So anyway, better choice is DG method.
Okay, thank you.
But to answer your question, what parameters? So it's whether normal or not normal. If it is normal, you can go for simple wash.
Okay, sir. Thank you.
Yeah. So is it okay to use alpha camotrypsin if a sample takes more time, 40 minutes in liquefaction? So for this, I have to check. I'm actually not authority to say this should be used or not, but we'll come back on this question from Isha Rajput. We'll discuss with the right expert and we'll get back to you on this. Chandan, you can note down this question from Isha Rajput. We'll have to get back. And if you can share your phone number or email, we'll get back to you.
Okay, so any other question, participants? If no, we can conclude this session here only. And we will meet again the next week with new topics, new audience, and new guest on the new session. Yeah, Isha, we will share that. Okay. What question do you have, 𝗦𝘂𝗶𝗺, that you are sharing your number? Do you have any question? Okay, I think we should conclude here, Sohan. I think we don't have any question now.
Okay, thank you very much. Thanks everyone for joining. Thank you,
yes, we will share the, stay with us. I'm just sending the link of certification that you will get it. Just give me 2 minutes time. I'm just sending the certificates. Okay. So you need to fill your details and you will get your certification in your email. And also I will request you just share your certification in WhatsApp groups, email, wherever you want to share. Okay. So here is the certification link. You need to fill your detail and certification will be released in your email. You can just share in LinkedIn, what's our status, so that social media, Instagram or Facebook. Here is the link. Just copy this link. You will get the certification here only.
Chandan, you note down the question from Mr. 𝗦𝘂𝗶𝗺. We have to get back to him as well.
Okay, just hold on. So, okay. Okay, 𝗦𝘂𝗶𝗺, we have noted down your question also. Yeah. Anyone, any questions more? You don't need to share your email ID. You will get, just I have shared the link. You just to go and fill your details. Certification will be shared in your email. Don't share your email ID here. I've already shared the link again. Wait, I will stop giving you access. So no host and co-host. Okay. I'm just sharing the link again. So you need to click on that link and your certification will be in your emails. Here is your certification. Please put thumbs up. You have received certification links in your chat box. Yeah, Arpita, thank you. Okay, so you have received. So just take the certification link. You will get in your email only. No need to share your email ID here. Okay, thank you. We have received Arpita. Okay.
Okay, we conclude the session now.
Yeah, we can conclude this session now. If you have
Okay,
any
thank you.
question,
Okay.
thank you. Yeah, thank you, Rishabh. You have received certificate. Thank you. You can just share in your WhatsApp everywhere. Thank you. I'm just dropping this session here. Thank you.